Let's say that after demonstrating that strains 2 and 3 are mutations in the same gene, at very rare frequencies, I observe wild-type flies when I cross them. How can that be explained? Well, it can be explained that these two mutations are structural alleles. While they are alleles of the same gene, the mutations are located at different sites in this gene. So consider this white-eyed fly. Let's say it's a heterozygote between strains 2 and 3, which were shown to be in the same gene. But let's imagine that the mutation causing the white eyes from the female parent is at the beginning of the gene, while the mutation from the strain of the male parent is towards the end of the gene in the second half. When the female makes gametes, and I'm saying female because it's sex-linked, but this would then be true of autosomal genes in either parent. When the parent makes gametes, crossing over occurs in meiosis. Now, crossing over would typically occur, say, between separate regions of a chromosome, but with very rare frequency, it could actually occur within the same gene. And so you could separate the beginning of a gene from the end of a gene and cross over and replace it with the complementary portion from the other gene from the other parent, in which case you would end up with one chromosome in one gamete that has both mutations, one in the beginning of the gene and one in the end of the gene, while a second chromosome, which is destined to a different gamete, which would have neither mutation. So therefore, it would have two wild-type sites. It would be a wild-type allele that would then cause red eyes. And so this very occasional occurrence of a wild-type fly would mean that the mutations causing strains 2 and 3 were not in the same site within the gene, and recombination could rarely separate them. If wild-type flies are never observed in these heterozygotes, then the two mutations would be concluded to be at the same site of the gene.